Hello, so today I'm going to be starting a new series on the fundamentals of molecular biology. At this time, I don't know how long the series is going to be, but hopefully by the end of it, you'll be able to have a very strong foundation in how life functions at the molecular level and get some intrigue or be able to use this for understanding how life works. So this is actually a very specialized branch of biology. It was popularized as a major in the 70s, I believe, where you're taking biochemistry and more structural interpretations of chemistry to focus on understanding how biological activity works at the molecular level. So you have these big macromolecular structures which you'll never see in like a general chemistry class. You're focusing on very small molecule things, but biology things are huge in comparison. And that's really necessary to facilitate a lot of these biological processes. And it's very interesting how life is able to construct these structures and also use these structures with purpose. So this is possible because within all cells there is a sequence of instructions that tells the cell how to maintain and reproduce itself, and that's called the genome. So what this really is information or instructions. It's a repository of instructions or data, and it has to be read and processed into transferable informations that can be manufactured to allow the cell to maintain life and also reproduce life. So that's what we're going to get in here today, focusing on the genome. So there are a couple elements of life, but some of the most important are carbon, hydrogen, oxygen, and nitrogen. And they can be used in various configurations to basically make everything you need for life. So carbon, of course, is one of the most special elements because without it, there possibly couldn't be life. So it has a couple properties that aren't found with any other element, which is why it's so honestly crazy that it even exists. So it can form four strong covalent bonds, and this allows various structures to form, like rings or other sorts of configurations, as you can see in this tetrahedral form. So it's a strong bonder, and it has a smaller atomic radius, so it doesn't get in the way of bonding and decrease sort of the energy or the strength of the interactions, hence the shorter bond length because it has a smaller atomic radius. And in terms of electronegativity, it's nonpolar, so you can have oils form from very long carbon chains that don't want to dissolve in water. That's why oil and water don't mix. They're mostly carbon and hydrogen. And because carbon is very flexible, actually, it can exist in many configurations, which gives life some flexibility when designing biomolecules. You can use different configurations to really fit and evolve into each other. So I make a big deal about carbon because in popular culture there is some emphasis on silicon based life forms. And to kind of put to rest some of the thoughts about silicon based life forms, silicon, although it's very similar to carbon, it can bond in a tetrahedral arrangement and it's in the same family as carbon. You can see it's right below it. It's not carbon. So the principal factor we'll look at is that carbon dioxide is a gas and it's a waste that the cell uses and it can just sort of poof it out into the atmosphere. Whereas silicon dioxide is rock, it's sand. So if a cell is based in silicon, it would have to somehow get rid of that rock, it wouldn't be able to have sort of this fluid environment. Like silicon dioxide is not soluble. So I really couldn't see how life could function if it was silicon based. Potentially, it wouldn't even have water in it. It would have to have something else to dissolve silicon dioxide. So that's possibly a very consequential chemistry thing because chemistry is the same throughout the universe. So if we never can or envision life with any other element that is like carbon, then carbon must exist to all life, and we can search other planets for their carbon abundancies or biological waste products to find life on other planets. So, speaking of building life, there are also some various hypotheses for how life started on Earth. So, before life, primordial Earth was literally a barren hellscape. There was lava and meteors raining down, and the early solar system, the primordial solar system, was very inhospitable place to be. Yet, it's thought that it contained all of the components necessary for life. For instance, as you can see in this picture, there are thermal vents providing heat, atmosphere protecting from radiation, and lakes and whatever forming. But in 1952, there was a very interesting experiment by Miller-Urey that simulated the conditions of primordial Earth by adding water and methane and some ammonia into a glass beaker, as you can see on the bottom right, and then shooting some electricity in it. And when they check it a couple days later, they find that a lot of the biological molecules we find in that you need to build even bigger molecules are forming here. So, it's kind of interesting. Like primordial Earth has a lot of lightning strikes or kind of churning things, churning the gases to make these complex molecules. So, that's very interesting that these could form with not a lot of care or precision. So, on other planets, if they have similar starting conditions to Earth, perhaps they could also have life, such as Earth. There's also another competing hypothesis from the astrobiology field, where they're observing on asteroids, out in space, they're finding the presence of these biological molecules on them. So their idea is that planets can be these barren hellscapes, but they can be bombarded by asteroids with these molecules, essentially delivering the components of life to planets, which could also be, very fascinating. But I'm not here to debate which hypothesis is right. I'm just here to say that there are just four main classes of biological molecules where if we find them on other planets, we can get a little excited that there might be life, which are nucleotides, amino acids, carbohydrates, and lipids. And for the rest of this introduction, we're going to focus on nucleotides specifically, of which there are five: adenine, guanine, thymine, cytosine, and uracil. And these are really the code of life, how we can put them in a sequence and compose the instructions for cells to function. So in 1928, there was an experiment by Griffith which demonstrated that some sort of hereditary information could be transferred from heat-killed bacteria to live harmless bacteria. The dead bacteria were harmful, and when you put them or put some of their essence, let's say, into live harmless bacteria, they would turn deadly, which is strange. He didn't know what the transforming element was. A lot of people thought that it was protein at this time. Some thought it was just arbitrary. But in the 40s, Avery, McLeod, and McCarthy showed that whenever you redo this experiment, but you destroy the protein, destroy the RNA, or destroy the DNA, you should get different results. But the destruction of the DNA stopped the transforming ability. So therefore, DNA is the hereditary element. It's able to be passed on, and you can transfer those instructions. In 1952, the Hershey-Chase experiment also confirmed that DNA is the genetic material. And more so talking about the structure of DNA, Chargaff observed in the 1940s that DNA, whenever it's paired up, there's obviously two strands of DNA that make one full segment of DNA, but the proportion of A's and T's, or adenine and thymine, is about 1 to 1, and the same with guanine to cytosine. So, therefore, adenine must pair with thymine in the 1 to 1 ratio, and the same with guanine to cytosine. This was helpful because in the 1950s, Watson and Crick were able to model and put together a structure of DNA on the assumption that adenine bonds with thymine and cytosine binds with guanine in an anti-parallel fashion, which we'll talk about in later slides, to form this double helix structure I'm sure we're all familiar with. But this is important to find out which nitrogenous bases bind to each other to form DNA, to form these instructions for life. So then we get into sort of composing the genome. So these special molecules are called nucleotides, and they utilize a sugar backbone, so like glucose, fructose, whatever. These are just another ose. So some common ones in the genome are ribose and deoxyribose. And the point to make here is that ribose would have two OHs here and here. This is deoxyribose. So it's literally ripping oxygen off of here and just leaving the one here. And that's the difference between ribose and deoxyribose. And the special part about nucleotides is that you need to add a phosphate group over here and also your nitrogenous base. So really, you can kind of envision how you can use these to build larger structures. So they can be polymerized into these longer strands, which can bind in reverse complements to form double-stranded DNA. I'll get into that later, what reverse complements actually means, but just focus on that, just the basic structure, that DNA can be compacted around nucleosomes because it's basically like a string, how you can stretch it, condense it, put twists in it, which makes it easy for life to sort of manage this very complex molecule. And it can be condensed in the chromatin whenever it's wrapped around nucleosomes, and then further condensed into chromosomes like this X shape we're all familiar with. So what we really need to focus on here in the terminology is that this OH group here on the deoxyribose sugar is located in something called the 3' position. So 1, 2, 3, 4, 5. This is just a reference to counting the carbons in the actual sugar molecule. So 3' says that there's something bound at the third carbon, starting at the first one right here. And the phosphate, of course, is on the fifth. So this is a pentose sugar. So moving on, the actual genome is constructed in a very specific fashion. So the DNA chain can actually only be extended in one direction due to the actual chemistry of the nucleotides. It will always build in that 5' to a 3' direction. So you have this phosphate group that I was showing earlier, and the big deal is that the phosphate can only attach to this 3' OH group. So this attaches to here, which adds another molecule and another exposed OH group, which can be further attaching, attached until you form this long chain, as you see here. So these 5' phosphate groups actually attack the free 3' OH groups and bridge the molecules together and extend one DNA chain. This is called phosphodiester bonding. So because DNA can only be made in a 5' to 3' direction, you have to have a complementary strand that runs anti-parallel, so you can only build it 5'3", so going 5'3" in the other direction is going like this. So they're anti-parallel to each other, and also it's a reverse complement. So as we established before, adenine binds with thymine, you would have to have sort of the opposite binding partner. So you would have to have cytosine here, guanine, thymine, and then adenine running in the opposite direction, which is also anti-parallel. So that's just one of the very fascinating things about DNA, how you can have such a high level of complexity and establish sort of information from this or order from this. Also, to note on this slide, the way these nucleotides are able to bond to each other is through hydrogen bonding, where guanine and cytosine have 3 hydrogen bonds together, while adenine and thymine only have 2. And it's significant because the guanine cytosine are, of course, stronger bonds because they have more hydrogen bonds together than adenine and thymine, which will be important later when we discuss the actual functionality of DNA. So, to also talk about the structure of DNA, its 3D structure contains major and minor grooves. So if you think about it, you have these 2 anti-parallel strands and you're trying to spin them together. There's this sort of asymmetry of the attachment of this sugar that causes some of these strands to be sort of tighter and some of these to be wider. And again, this is because, as you can see in some of these, that these sugars are anti-parallel, and that sort of puts a kink in your DNA strand. And the effect of this is that you have this major groove here, which is really important because you're exposing this information held in the DNA, which can be read by other proteins. So this is how the cell can actually read the DNA without opening it and sort of stick some proteins in there to facilitate biology or using the information. And this is my last slide here. So where does the genome actually live in the cell? So the cell stores its DNA in a nuclear envelope, or the nucleus, in the center, usually, of the cell. And this segregates DNA-related functions from the rest of the cell. So there is something about the DNA that you need to be careful with it and get it just away from everything else happening in the cell and put it in some special enclosed space in the center. So just another tidbit, sometimes you'll see, or just imagine always, DNA is those X-shape chromosomes. That is a super condensed form of DNA. As you can imagine, if it's all condensed like that, it would be very hard to actually use something like that. So you want to have it decompressed usually. So it'll exist kind of as spaghetti within the cell, and it'll condense into those X shapes whenever the cell is ready to divide or replicate. So that's the end of the introduction lecture. I hope you learned something. Tune in next time. Thank you.